Okay, thank you, Natalie, for this very kind introduction. So I actually changed the title a little bit of my presentation. I tend to do that all the time. But the reason for that is that I would try to discuss today with you that we might want to stop thinking about FSHD1 and FSHD2 as two separate disease entities and actually start to see them as opposite extremes of a disease spectrum. So this is, I'm based in Leiden in the Netherlands. This is let me go back. Well, is this? Okay, this is our research building on one of the few, the rare sunny days in the Netherlands. And our institute has been working on FSHD for almost 3 decades now. Started in the 80s with Professor Padberg on the clinical side and Padberg on genetics. And since then we have always been involved in the genetic studies for FSHD, the molecular and cellular biology, but we also do the DNA diagnostics of FSHD and also assist in the diagnosis of many patients worldwide. And here you just see an example of all our blood DNA samples that we have and the different procedures that we use to diagnose the patients with FSHD. Now, being for so long time in FSHD also makes you realize that actually in the past 5-6 years ago, we have really seen a complete change in the direction that our studies are taking. So until 2010 or so, everyone in the research field was really dealing with the question, what is causing the disease? How can it be that this contraction of this repeat, this D4C4 repeat, leads to a muscle disease? And after the research field identified this mechanism, really what we see now is that we realize we know enough about the mechanism. Sure, there are still many things that we need to understand better, but we know enough to start thinking about how can we actually stop this process or maybe even reverse this process. And to explain that better, I'm going to use a couple of slides on basic biology that may be a little bit redundant after the many other slides, but maybe somewhat useful for my presentation. So the two things I want you to remember is actually there are two basic principles within the biology that are essential for life. The one is called replication. This is basically the copying of our DNA so that the information, all the information is stored in our DNA, is faithfully copied to the daughter cell so that each cell exactly has the same information to work with. The other one is what we call transcription and translation, and it's basically using the information that is embedded within the DNA. So in the DNA are our genes, as you understand, as you've heard before, and actually these first needs to be copied into a molecule which we call RNA, and then it will be translated into the proteins, which are the building blocks of our cells. Now, this is not a simple task for a cell, and this already has been alluded to earlier, so also I think we have 2 meters of DNA, not 3 meters, but maybe Italians have 3 meters, and that's why they are so sophisticated. But anyway, whether it's 2 meters or 3 meters is not really important. Putting 3 meters or 2 meters of DNA within a single cell or a single nucleus is the equivalent of taking a rope the length of half a marathon and wrapping that up in a tennis ball. So this is an enormous challenge to the cell, to the nucleus. And the way it basically does that is by really packing it very tightly around proteins. And I always make this comparison with this spring. So basically the DNA is wrapped like in very tight coils, like you see over here. But if it's wrapped this closely, you can't use the information that is in there anymore, right? So basically what the cell can do is open specific parts, making it a little bit wider, a little bit opener, so that it can start reading information that is in there. And actually, that's also what you can see over here in a more complicated way. Here we have our DNA wrapped around proteins, and basically the cell can make decisions on which parts of the genome it wraps very tightly, so to make sure that there is enough space in this nucleus to put these 2 meters of DNA within the nucleus, and which parts it opens up so that it can use that information that is available there for the cell. And it really makes sense because we have only one genome. All our cells have all the same genome. Yet we have 11 body systems and as much as 37.2 trillion cells. So, but we all know that a blood cell is completely different from a muscle cell. So the blood cell needs to use completely different information from the DNA than the muscle cell. It doesn't make sense for a blood cell to use the information that is necessary for a muscle cell, and vice versa. So that's why the cell is basically capable of only using the information that is necessary for its life. Now, and this is what goes wrong in FSHD. So normally this repeat is long, and it is in this part of our genome, tightly wrapped. The information cannot be read. In the most common form of FSH, FSHD1, this repeat gets shortened to 1 to 8 to 10 units, and then actually it becomes in a more open configuration. So suddenly this information that is embedded within the repeat can be read by the muscle, and it's producing this protein DUX4. And we've heard today already that this DUX4 protein is not tolerated by the muscle, and it basically damages the muscle. So this is FSHD1. Now I also told you that you need proteins to keep the DNA either in this configuration or that configuration. And one of those proteins is called SMCHD1. So this SMCHD1 that binds to the repeat and is necessary to maintain the repeat in this closed configuration. Now patients with FSHD2 have a normal repeat size, like in control individuals, but they have mutations in SMCHD1. This reduces the activity of SMCHD1 on the repeat, and therefore the SMCHD1 is not capable to maintain it in this repressed, in this closed state. Therefore the repeat also becomes in this more open configuration, and the consequences are identical, that DUX4 is being produced. Recently we identified another protein. It's only in a very few patients, but also this protein in this case is not necessary for the maintenance, but basically for the establishment, for putting it here in this part of the spring. And if you have mutations in DNMT3B, like with SMCHD1, it leads to reduced DNMT3B activity, and hence the repeat ends up again in this part of the spring with the expression of DUX4. So we have, I think, a very plausible, uniform disease mechanism for FSHD1 and FSHD2. But it doesn't explain the clinical variability of the disease. And this is already known from the first description. Here are the two first patients ever described with FSHD by Landouzy and Dejerin in 1894. And these are two, I believe these are two nephews, and you can already see there's a lot of clinical variability between them, as you know from your own families. So we are really interested in what causes this genetic or this clinical variability. Why is one individual with the mutation a non-penetrant gene carrier, while the other is severely affected? And I'm particularly interested in this, as already was also mentioned earlier today. I'm particularly interested in those individuals that do have the mutation but not present with the disease, because there may actually lay some answers in how we can treat this disease. What is it that makes these people being healthy instead of affected? And of course, now we have identified these proteins that are necessary to keep the repeat repressed. We were wondering whether those actually might be these long-sought modifiers. And I think, again, from our families, studies with our families, we are actually finding some clues to this genetic or this clinical variability. And I'm showing here just one of those families that we have analyzed, but we have now many more, where we actually recognize that there is a co-segregation of FSHD1 and FSHD2. That means in this case that this gentleman over here actually has a contracted repeat. So he has FSHD1. The mother, or the lady over here, has a mutation in SMCHD1. So she is a carrier of FSHD2. And if these two mutations come together, and that can happen because the one is on chromosome 4 and the other one is chromosome 18, these individuals have both FSHD1 and FSHD2. And individuals with FSHD1 and 2 are much more severely affected and a very rapid progression of the disease. So you see a synergistic effect of the two mutations. And that's what I'm showing over here. So here again, the repeat is closed, it's tightly wrapped. In FSHD1, you get an opening causing the production of DUX4. In FSHD2, by mutations in SMCHD1, you get the same phenomenon, but in FSHD1 plus 2, you basically see the contraction and the mutation leading to a much greater opening of the repeat, much more production of DUX4, and a much more severe phenotype. So these genes for FSHD2 are modifiers for FSHD1. Now, one important observation that we did also with support of FSHD Global is reversibility, because of course, now we know that this is the problem in FSHD, whether it's FSHD1 or FSHD2, it doesn't matter. It's this opening of the chromatin, or this opening of the DNA structure and the production of DUX4. Can we actually reverse that? And yes, in tissue culture, so in cultured cells from patients with FSHD1 or FSHD2, when we add SMCHD1, we can actually close the repeat again and prevent the expression of DUX4. I think this is very important information. Yes, it has been in tissue culture, in the laboratory with protein, adding extra SMCHD1 to the cells, so we are far from the clinic. But it tells us that in muscle cells, this is a reversible situation. So I started my talk by discussing with you that I think we should stop talking about FSHD1 and FSHD2 and just consider it as a disease spectrum. And this is what I'm trying to show over here. So basically what we're starting to see here is that if you have very short repeats, then the repeat by itself is the major contributor to the production of DUX4 in the muscle. But when the repeat starts to increase in size, it is not only the size of the repeat or the contraction, but it is also the addition of modifier loci, so like SMCHD1 or DNMT3, natural variation that occurs in those genes that contribute to the probability of disease presentation and progression. Now finally, how about therapy? I think with the mechanism that we know by now and the action of DUX4 that we are realizing, we are in a position to think about different ways to pursue therapy in FSHD. And as I already said before to some other people, I think that I have always been very skeptical about a therapy for FSHD, but now having this in hand, it might be that FSHD is actually one of the more treatable diseases amongst the muscular dystrophies. And the reason for that is that we do not have to put something back in the cells, which you typically do with monogenic diseases. But basically, the only thing that we have to do is take something away. We have to prevent that DUX4 is being produced. And we can do that in different ways. We can think about, as I showed with the SMCHD1 addition, that we actually put the repeat back in its original place in the spring, so that it cannot read the information anymore. The other possibility is that we might actually prevent the production or destroy the production of these RNA molecules, like Yi Wen showed with the antisense oligonucleotides. The other possibility might actually be that we prevent the production of the protein. And finally, we might actually also prevent the action of the DUX4 protein or one of its downstream targets. Well, this is probably more problematic because we do not know which of these pathways, or maybe all of them, are contributing to the disease. So the higher up in the hierarchy, I would say, the more likely will be a feasible therapy for FSHD. And with that, I would like to also put up my acknowledgement slide with a great bunch of scientists that are working in my group and my collaborators, and of course all my funding and my support, including FSH Global. And I thank you for your attention.
So you mentioned how a person can have FSHD1 and FSHD2. Is there, and you said the only way to identify that is because they have more severe symptoms. Is there a testing that you can do to find out if you have both 2 and 1?
Well, so it was actually the rapid or the early onset and the very rapid progression in these familial cases that triggered the neurologist Sabrina Sacconi in Nice to ask us about whether this could be the presence of FSHD1 and FSHD2 in this individual. There are tests out there that we can easily do nowadays, and I think also with the previous presentation on the genomic sequencing, actually that would actually immediately tell you whether there's FSHD1 and FSHD2. So I think we will make great advancements there in the near future.
If the person in question is like earlier we said 1 in 3 people with FSHD are just sort of sporadic. They don't have any family members with the gene. So would they not count in this case of FSHD1 and 2?
Oh, that's still entirely possible.
Oh.
Absolutely, yeah.